Okay, so we're going to test this theory that it's going to let me do things. There we go. Alright, so what I'm going to cover in this video is two-way independent ANOVAs, or basically between subjects ANOVAs. And I'm re-recording this lecture, so it's not in a loud classroom this time, because we had a bit of trouble with the syntax for this, and I'll talk about that later in the video. So first, what is a two-way ANOVA? What we did last time was we covered one-way ANOVAs, so one independent variable. And so anytime you see the word way, what you're actually saying is how many variables there are. So a two-way ANOVA has two independent variables, and this could be between subjects or repeated measures, but right now all we're doing is covering between subjects. A three-way ANOVA has three independent variables, and by using independent in the sense of research design, we're talking about the participants are in all different conditions. So independent variable also means like the predictor variable, but if we say the ANOVA is independent, what we're really meaning is between subjects, where everyone is in a different condition. And remember that conditions are the combinations of levels. Several independent variables is known as a factorial design. And so sometimes people will call this two-way between subjects. Okay, that tells me that there's two IVs and they're all between subjects, which is nice. You can call it a two-way factorial ANOVA, but that's really redundant because two-way automatically makes it a factorial design. Or I can call it just a factorial ANOVA, which is probably the least amount of information that you could get in this scenario. We could also do two-way repeated measures ANOVAs, which indicates that all of the IVs are repeated, so everyone is in every condition. We could do a mixed ANOVA, which is one independent variable is between and one is repeated. So the best way to talk about ANOVAs is to use this sort of notation that I'm going to talk about here, which is that we have the number of levels times the number of levels. And then tell me what type it is. So this is the indicator of how much information we can cram into three little numbers. So what that tells me is that one IV has two levels. The other IV has two levels, which altogether makes four conditions. So it's a 2 by 2, right, like this. And then all of those IVs are between subjects. I could also have a 2 by 3. It doesn't matter which number you put first. But this is the way I think is the best way to talk about ANOVAs because it's the most informative. So it tells me how many levels in each IV there are to expect coming up, and then also it also tells me how many conditions there are to think about post hoc tests. So what are some of the reasons why we might do a factorial design? Well, mostly that is all about how the variables interact. So I don't think you would do a factorial design if you weren't interested in the interaction. And so we're really going to heavily bias talking about post hocs due to the interaction. Because if you didn't care about how they interacted, why are you testing them together? Why don't you just test them separately? So the interaction, what is an interaction? Well, it shows how the effect of one IV depends on the effect of another IV. So it is usually more interesting than the main effect. So in our little fake study here, we have gender and sport. And so we asked students how they felt about different sporting events on campus, given what gender they are. So do girls rate sports differently than men? And so that effect of IV, the effect of gender on sport rating, would be an interaction. And it's not just women rate sports higher than men or women rate sports lower than men. It's women rate different sports differently than men, we hope, in this interaction. And so it's using both variables at the same time. So essentially there are differences in condition means rather than differences in level means. Okay. And so to get into how do I test one of these things, is that all on this slide? Okay, good. Just a reminder about the assumptions for ANOVAs. So they're the same as one-way ANOVAs. You still have to check accuracy, missing data, and outliers. There's still only one DV, so there's no reason to check correlations because your variables, your continuous variables are not going to interact with each other. And then additivity also does apply to the IVs, but when you set up a ANOVA, it's sort of hard to make them non-additive. The real problem becomes when you have unequal sample sizes in each cell. If they're not additive, then you're missing half the cells. So it's a little bit harder to have additivity problems with ANOVA. So you want it to be normal, linear, homogeneous, so using Levene's test, and homoscedastic. So remember that when we're talking about IVs, each IV has its own set of levels, and then the combinations of levels are conditions. So the interaction is all about conditions. The main effects, which is kind of a new term, are about the levels, and the interactions about the conditions. And so we're going to talk about doing interactions that's either across or down, but not both. And we'll talk about that more in a little bit. So for this example, we've got gender of a participant, a male and female. So the IV itself is gender. Okay. The levels are male and female. 3 sports, none, volleyball, and football. So this is sport. And the levels are none, volleyball, and football. And then the conditions are who's rating what. So males rating the none condition, males rating the volleyball condition, males rating football. You get the idea here. Females none, females volleyball, females football. Okay, those are the conditions. This DV is satisfaction with athletics. Okay, so these are the sports that they attend. So we found people who only attend one sport or no sports, and then asked them how satisfied they were with athletics. Okay. Oh, I just did this, but you get the idea here. So more room this time. Male, female, here's gender, none, volleyball, football, here's sport. Okay, and then these are the interactions. So what do I mean by across or down? Just as a preview. I can analyze for the post hoc test across, or this would be down, I guess. So down, and that would be 3 tests. So this would be male versus female none, male versus female, oops, I don't know what I just did, female volleyball, male versus female football. Or I could compare going across. So none for just males, none to volleyball, none to football, volleyball to football, none to volleyball, volleyball to football, none to football. And so that would be 6 tests. 3 tests here and 3 tests here equals 6 total. So 1, 2, 3, 4, 5, 6. Compared to my 3 going down. Not very often do you want to compare like male none to female football, because those have nothing in common. Except they happen to be in the same experiment. So usually you'll pick across or down. If you aren't sure which way to go with your hypothesis first. The second answer is to go with the lower number of tests. So there are only 3 tests total here, so I would want to compare down. Okay, so what we're going to talk about now is more theory-based. So we're going to talk about how does the variability break down. Okay. So we're going to start with sum of squares total, which is what we did last time, and talk about how that breaks into sum of squares model and sum of squares residual. But then model is now going to have 3 new pieces because it's not as simple as one IV anymore. So I sort of have a chart here, and it looks crazy at first, of how all of this breaks down. And I'm going to walk through each one of these things one at a time. But essentially, we're going to break into how are all these things calculated. So sum of squares total first. And so sum of squares total is me, my individual person, minus the grand mean. So that tells me how much variability in the entire study there is. So how different am I from the grand mean, and ignoring all of the groups, which will give me a total amount of cookies, to go back to my example I used in class, is how many cookies are there to hand out to people, and then where are those cookies going to go. So we've got just the total number of variants in the study, DF total is the number of things that we're using. So this is big N for the number of people, minus 1 for calculating the grand mean. Okay. You don't really use DF total very often, but it's a thing. So here's the formula. For calculating it, you could do this by hand if you wanted to on our data set that I have included, but there it is. So SS model is my group mean. Okay. Now I'm in 2 different levels. So remember, this condition is a level plus a level. Okay. Minus the grand mean. And what we did last week was just my group mean minus the grand mean. So it's actually the same idea, but now group has a different connotation. So group could be gender, it could be sport for main effects, or it could be gender and sport. So we have to kind of break this down differently. So by using my group mean and talking about it being just levels, ignores the fact that these variables are structured. Okay. And so they're structured by condition. So now we need to figure out a way to account for the fact that we have 2 variables and this interactive structure. Okay. Oh, and then here's the formula. Let me erase this real quick. Okay, and that's the old formula. So now let's talk about just gender. So the formula for just IV1, if we just, sometimes this is called sum of squares A, just because it's the first variable, but you could think about it as sum of squares IV1 too. So it's the same formula as SS model, but now we're just going to pretend like this is the only variable in existence. So it is the level mean, so gender's mean, minus the grand mean. Okay. And the degrees of freedom for that is the number of things we're using, so K, which is the number of levels, minus 1 for the grand mean. Okay. And so that's just how this formula is working here. Level mean minus GM. Okay. And so that's actually the same thing as a regular one-way ANOVA, but that ignores the other variable. So let's add that variable in. So now for sum of squares IV2, it's the exact same formula, but now we're going to do sport minus GM. Okay. So we've done gender minus GM and sport minus GM. Now, what do I do about the interaction? Okay. Let's talk about marginal means first. So anytime you have your design here, okay, we're going to, oops, that's an F. We're going to talk about the marginal means. And so what that means is it's the means in the margin. So this is just males' overall rating and just females' overall rating, just none, just volleyball, and just football. Okay. So this is left over from the days when we actually had to calculate everything by hand. And so level means are called marginal means because they were written in the margins, whereas these here are condition means. Okay, so those are the condition scores. Okay. And so all a marginal mean is that if the other variable didn't exist, here's the score for this level. Okay, so here's the score for males, totally ignoring sports. Here's the score for none, totally ignoring gender. So now what to do with the interaction? Well, there's a way to hand calculate it other than this, but it's pretty gross. So basically what happens is we're going to say here's gender, here's sport, because those are fairly easy, and then everything else left over, we've divided up our little cookie jar here. Here's gender, here's sport. Now the rest of this goes to interaction. Because we have a sum of squares total, right? And a sum of squares model. So we know how to break things up. Okay. So the cool thing is that sum of squares residual, or error, also doesn't change. It's the average variance across group. But now, again, I've got to account for the fact that my group is not just male, not just football, it's now a condition. So it's each person minus their condition. Okay. Which is the basic idea of a variance formula, right? Squared. So it's the sum of all the differences from conditions, so it's the average variance across groups. And so to end, let's go back and look at this. So sum of squares total, we did mean minus a grand mean. How different am I from the overall average? And this is total variance. Okay, it's the sum of squares total. Then I broke that into SS model, which is these three pieces here. So I have two main effects, 1, 2, and the interaction. Okay. So how different is my level from the overall average? How different is my level? So my level minus my grand mean. Okay, and that's an average level minus the grand mean. And then how much of that difference in conditions is due to the interaction, not just the levels. So how much of the differences in conditions is due to the interaction? Okay. It might be how much are due to the interaction, but that just sounds funny. So how much of these two things the SS model is left over after accounting for the IVs? Okay. Residual is how me minus my condition. How different am I from my own group? So remember, variance within group in a between subjects ANOVA is error. We don't know why it's happening. So we did levels minus 1, levels minus 1. So the interaction degrees of freedom is these two things times each other. Because both of these lead into the interaction. They're both part of the interaction. And the residual degrees of freedom doesn't change. So it's n minus 1 plus n minus 1 plus n minus 1, which is essentially this stuff right here. And then the only thing that I don't have covered really in my slides is the rest of the table, because this table is so similar to the other tables that we've talked about. But mean squares, right, is calculated by dividing across. So sum of squares divided by degrees of freedom, going this way. So it divides going across. And then F is my mean square model always over mean square residual. So this is the denominator in all of these things. Okay. And so we just take mean squares divided by the residual for all of them. So it's still a ratio of good variance to bad variance. Of variance for just gender over error, just sport over error, and just interaction over error. Alright, so a little bit on how to do this in R. Give me just a second. So one thing we're going to do is we're going to use the star operator. And that, unsurprisingly, tells it to multiply. And so this should be when you multiply variables together, you're creating interaction. And then what's called the lower order effects. What I mean by lower order effects is the main effects. Okay. So main effects are just one variable at a time. The higher order effect has two variables at a time. So lower order means there's less variables involved. Higher order means there's more. So if I include this in my R code, it automatically creates these two other ones for me. So that's nice. It's a little simpler. Alright. Since we're doing between subjects ANOVA, we still need to check for homogeneity. And so we're still going to use Levine's test in the CAR library. And here's what I mean by the multiply operator. So it's x times x, data equals data. Remember, center equals mean if you're using a traditional ANOVA, or it will do a median center, which we don't want. To run the ANOVA, it's the same formula, x times x, data equals data. So let's see if we can get this to run for us in R here. Plug in my little keyboard. So let's get PowerPoint to quit. Here we go. Alright, keep recording here and go to Studio. Apparently did not decide to exit here. And there we go. Okay, so I have my syntax pulled up here in RStudio. And since I'm doing this on a different computer than normal, I'll set my working directory. So I'm going to import this file. And what you'll see is our dataset file has gender, and it imported this little goofy eye symbol, so we're going to fix that here in just a second. And it's imported as male and female, sport as football, none in volleyball, because that's alphabetical order, and then satisfaction with sport. I'm going to turn off scientific notation. and that will allow me to just see the p-values as they are, not in SciPy. But the next thing I'm going to do is kind of fix my gender column here. So I can do that with the call names function. Okay, and let's do data set. Okay, square brackets for the first column equals, and if your data set imports this way, you will want to fix it or the rest of this code will not work. Gender. Okay, and so you'll see up over here that fixed my gender problem. Okay. So I'm going to check the data set real quick, just make sure it looks okay. So I've got 24 people in each gender condition, 16 people in each, level, 16 people in each sport level, and then my satisfaction variable, let's say it runs from 0 to 100. Now one problem I have is that this imported in alphabetical order, so I'm going to change this to be none football and volleyball, because that, like, picture-wise makes more sense to put none on the left. So all I've done now, if I rerun my summary, is just reorder that factor. So now it's in a different order. Oops. Okay, so I'm going to run, I'm not sure if car is on here, let's try it. Okay, I guess so. So we're going to run Levine's test. And so here's Levine's test, and it tells me that is not significant. So if you look at this p-value down here, 0.20, that is not less than 0.001. So there are no significant differences in homogeneity by condition. So this tested it for 6 conditions. Okay, so 2 by 3. Next thing I want to do is run my ANOVA. So I've got the same setup here, satisfaction. So y is approximately x by x. And then I'll get the summary. Let me go back to PowerPoint here. I could talk for a second about that. So, my quick warning is that there are different types of ANOVA math. There's at least 4. So type 1, type 2, type 3, type 4. Aren't those great names? Okay. Type 3 is the most common sum of squares, and that's what we've been learning. And you'll see, like, if you run an ANOVA in SPSS, that's the type you get automatically. Okay, and most people don't even think about this because it's so pervasive to use type 3. Type 1 changes if you change the order of the variables. So if I wrote gender times sport and then switch them, sport times gender, I would get different effects, because instead of controlling for the main effects and then testing the interaction, it controls each main effect one at a time and then does interaction. I know that sounds the same, but what'll happen is if one variable has way more variance than the other variable, it'll kind of eat the other one. And you'll see this happening in regression sometimes. If you do a hierarchical regression, where things happen in steps, that's kind of how type 1 works. If you are reading the Anyfield book or looking at any internet source ever, what you'll see is that they suggest using the ANOVA function, not AOV, but ANOVA, like this. Type equals 3 to get the type 3 sum of squares, and that is in the car library. That is perfectly well and good, but the ANOVA function with capital A assumes that your levels are ordered, which is not super common. That's the sort of thing that you want to be concerned with if you're doing a trend analysis. And so there are ways to turn off the ordering, but it's actually more complicated than just running the summary function. If you're reading these sources, it says the summary function is type 1. But after testing this for a bit, it's actually clearly type 3. It gives you the same results as SPSS. So I'm sure something has been updated somewhere that the books haven't caught up to, so you can just use the summary function to get type 3 sum of squares, which is what I did here. So now I have 3 values. So I have 3 different F values, 1 for each effect, and then my residuals here. So R does not give you total, but you could add them up to get total. So I've got everything for gender here, everything for sport, and gender by sport. So I'm mostly concerned with this column, right? So gender is not significant, but sport and gender by sport are significant. So how do I write those up? Well, let me go back here. So you want to use 1 degree of freedom and 42. So this 42 is going to get a lot of work out of it. and then 2 and 42, 2 and 42. Okay. And that's where I got these numbers in each one. Remember, all statistical letters are italicized. Then I pulled F from the F column here. So I use these numbers for F, and then these numbers for P. Anytime you have 3 zeros in a row, you go less than 001. Which, if I can get the swipe going, there we go, is how I got these numbers. So let me talk about effect size, and then I will move into what do I do with these numbers. So since we have 3 effect sizes, I can't really use eta. So remember, eta is sum of squares model over sum of squares total. I could do that, but now I have 3 sum of squares. So I could do it for each one, but the way eta works is it's the overall variance. And so that's not really what I want. Instead, we're going to do how important is each variable one at a time, or each effect one at a time. So we're going to use partial eta, which is sum of squares model over sum of squares residual plus sum of squares model. If you only have one variable, those two are equal, which is why I didn't talk about partial eta last week. But if you have more than one variable, you're leaving out the other models. So sum of squares gender over sum of squares residual plus gender, sport over sport plus residual, interaction over interaction plus residual. We'll talk about omega here in a second. Okay. Keep recording, I'm not done. Okay, so here we go. So to get those effect sizes, there we go. What I did was from this ANOVA here, I just typed in sum of squares residual, saved it so I could use it 3 times, residual, 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 and then I took the sum of squares for each one, gender, sport, and then the interaction, and just type them into the little formula. So be sure you include your parentheses here, so otherwise it will divide first, and you'll get something over 1. If you get a score over 1, you've done something wrong. Oops. Okay, so I've got 0.05, which is definitely small, 0.49, which is large, and 0.36, which is large. Right, and that is what I did here, was I just typed them in. But let me explain partial eta. So the effects are estimated based on the effect and the error. So it has a different denominator than regular eta, which is what I have here. So that denominator is a different one because it excludes the other effects. Okay. With one IV, partial and total are the same. With multiple IVs, most people don't ever report a total. Talk about, here's how you'd report them. So you're including, oops, I'm not in, sorry. This mode. So I would report those. Now let's look at partial omega. So partial omega is kind of a disaster for ANOVAs, but here's the formula. Okay, so this would be just for A. And let me do this real quick. So this actually right here is DF model for the one you're interested in. Because it's levels minus 1. That's essentially K minus 1. This here is a tricky part because you're doing A is the number of levels, and N assumes that you have equal people in each group, which is kind of problematic in the real world because that doesn't necessarily always happen. But you can always use the harmonic N, which is the average N across all of your groups if they aren't equal. A lot of these formulae are based on a perfect world with perfect number of people in each condition, which, as we all know, real research isn't always that perfect. Okay? That's why people favor type 3 sum of squares also, is because it tends to work better for unequal groups. Okay. And so I could fill all of these things in. So let's do that. Yes, keep my annotations, keep recording. Let's go back to Studio here. And so what I did to calculate omega is the first thing I did was run some tables just so I could see how many people there were. So that first table tells me the number of people to put here for gender. The second table tells me there's 16 people in each group, so for sport. Now what I did for the interaction was I coerced t-apply into giving it to me. So I said calculate the number of people using gender and sport together. And so that's 8 people in each condition, and so that's how I got this last number out here. And so the rest of this I filled in from my table using the right types of parentheses to get the right numbers. Remember that these should always be slightly less than your etas, your partial etas. So 0.02 versus 0.05, 0.44 versus 0.48, 0.31 versus 0.36, so pretty close. And so those would be the partial omegas. Remember, partial omega is supposed to represent the effect size in the population better than partial eta. And so what I want to do is graph these effects just to see what's going on. And I included the ggplot code to make these graphs, but I'm just going to show you the graphs. It's a little easier. So for gender, we said this was non-significant. So look at this. Here's mean 1, here's mean 2. There's really no differences between those two means. So not too surprising that this wasn't significant. Also, the confidence bars overlap. And so if we were doing confidence interval testing, we would find the same effect. If you look at sports-related ones, there's probably an effect here and an effect here. So football seems to be less than the other two. But across here, maybe not so much, because the confidence intervals are the same. So that's the main effect of sport all by itself, ignoring gender. And then now let's look at this bad boy. So this is the effect of them together, and here's why we have an interaction. This difference is positive. This difference is negative. This difference is probably nothing, but also positive. And so you get an interaction anytime you have a different pattern of effects across variables. So if I also could think about it this way. So women are rating them the same pretty much no matter what. Men have this big dip in their ratings. So we get this different pattern across variables. That's an interaction. So I'm going to end here because this is the end of this example. I'm going to take a quick break and then do part 2, which talks about how can I see interactions in graphs. So I'll draw you some more graph pictures. And also, how do I analyze this, the post hocs for the main effect and post hocs for interactions.